Hello. The charging of tRNA is an important aspect when it comes to translation, and this is mediated by the enzymes called as tRNA synthetases. tRNA synthetases have a big task because there are a large number of RNA molecules, out of which they should be able to bind specifically to only the tRNAs, and out of the tRNAs they should only bind to specific tRNA. And on the other side, there are about 20 different types of amino acids, and the tRNA synthetase should pick the correct amino acid, pick the correct tRNA synthetase and charge it. Amino acylation of the tRNA happens, and it means that there are two levels or two distinct parts of specificities that tRNA synthetases make happen, and that is how they mediate the charging of tRNA synthetases, charging of the tRNAs. There are about 20 different amino acids, and there are 64 codons. Out of the 64 codons, 3 are stop codons, so we have 61 different sense codons. And of course, there are multiple codons which code for the same amino acid, owing to the verbal concept. So how many synthetases would we have? One synthetase adds one amino acid to a group, or more than one, can add to more than one type of tRNA. So we can expect a typical bacterium, for example, to have about 20 tRNA synthetases. They are specific for the amino acid that is being added to the tRNAs. And there is also a convention about how people write this. Glue, tRNA, and superscript we have as GLN. Normally we would let's say the glue here, GLU, stands for glutamic acid. It's not for glucose. People tend to be confused as glucose sometimes. For glucose, the code is GLC. GLU is for glutamic acid, and the prefix mentions it defines what amino acid is going to be attached to the or charging the tRNA. The superscript is about what type of codon the tRNA recognizes. So basically, you cannot change this. tRNA GLN, if you meant that tRNA has a stop codon, has an anticodon that recognizes the codon for glutamine. GLN stands for glutamine. And here we have glutamic acid. This tRNA is charged with glutamic acid. So it sounds wrong, right? That we would preferably have the GLU, tRNA, and GLU, which would mean that glutamic acid is added to a tRNA that would recognize the codon for glutamic acid. That makes sense. Here we are going to have a little bit of a contradiction, and then we'll see how it happens. So the tRNA synthetase has a serious degree of specificity it has to have, which would mean that it should be able to differentiate between glutamine and glutamic acid. The difference just being this additional amino group that is present in the glutamine, and yet they can be present. And similarly, they can also have, say, phenylalanine and tyrosine. The difference is not big at all, and yet the tRNA synthetase, the particular one, can recognize if there is this OH present or not. And in the binding pocket, whether the OH can fit or not. OH is a charged group, or it can have interactions which may not be allowed into the binding pocket of the amino acid. Similarly, we can also have valine and isoleucine. They differ just by one methyl group, that can also have spatial constraints. So there are, what we should understand is, into the binding pocket for valine, isoleucine cannot fit because isoleucine is bigger. But into a binding pocket of isoleucine, valine can fit. And that is also can fit in the sense that there is a possibility for isoleucine aminoacyl tRNA synthetase to make a mistake of by having valine in the binding pocket. But there are mechanisms of steric hindrance, of spatial constraints, and also binding of the interactions of the amino acid with the binding pocket that will be able to distinguish from amino acid to amino acid, and eliminating the larger ones, eliminating the ones that do not make proper interactions and steric hindrances and other things. So that is how the binding pocket of the amino acid in the tRNA synthetase can eliminate any wrong amino acid that might be present. tRNA synthetases also have, say for example, if this is the tRNA synthetase, they have a way of touching or the interacting with the anticodon loop as well as the acceptor loop, and that is how they recognize whether the proper tRNA has come into the place or not, and that is how they can eliminate any wrong tRNA that might have come into the active site. So as we discussed here, tyrosine and phenylalanine, for example, so the favorable hydrogen bonds, because of the hydroxyl moiety, will allow the discrimination between tyrosine and phenylalanine. Isoleucine is again, as discussed, it's about the sterical exclusion of isoleucine. And there is also one another pocket called as the editing pocket. That is, once the proofreading, once the charging has happened, the editing pocket can recognize if the the product that has formed is proper or not. And it's all as is the case with all the enzymes, it's all about the interaction of the reactants with the enzyme and also the products with the enzyme. When either of them is not right, the reactants are not right, they will be eliminated. If the products are not right, they can be a reverse reaction can happen. So that way one can expect that there are multiple methods of how proofreading happens when it comes to addition of or charging of tRNAs. It is an important thing, because if there are large number of mistakes that would happen with regard to charging of tRNA, there is no reverse reaction to this. Once charging has happened, the ribosome would pick up the amino acid the charged tRNA and would add the amino acid, the growing peptide onto it. The ribosome does not have any discriminatory power. All it can do is it can only bind to tRNA, that is charged tRNA. It does not have any other discrimination. Of course, ribosome is moving on top of or reading the mRNA. It will come in contact with different types of the codons that are on the messenger RNA, and so it cannot have specificity to accept a particular type of mRNA, to proofread whether the charging of the amino acid to the particular tRNA is correct or not. There are one aspect here is that, say in situations like this where glutamic acid is added to a tRNA, in some species, bacterial species, there are the tRNA for glutamine is not present. The synthetase, tRNA synthetase for glutamine is not present, meaning that there is no way of a tRNA for glutamine, GLN, to be added directly with glutamine, because that particular tRNA synthetase is missing. So instead, what happens in that case is that first there will be a glutamic acid added to the tRNA synthetase that is specific for glutamine, and then the carboxylic group of the glutamic acid is added, aminated, which means now the glutamic acid has changed to glutamine tRNA for glutamine, right? So wherever the deficiency for tRNA synthetases could be in some of the species, in that case these kinds of counter or bypass the deficiency, bypass mechanisms for this kind of deficiencies are also there. So if we had two different tRNAs, GLU added to tRNA for GLU, so the glutamic acid here will not be acted upon that enzyme, aminated. Whereas if glutamic acid is added to an enzyme like glutamine tRNA GLN, that glutamic acid is aminated. So this is not a general case, but as is with biology, there is too much diversity, and in different organisms we may have different systems that may have evolved. See you later. Bye bye.